We have just seen that the interest of cells and animal model, and now we move to the last scientific session on stem cell and induced pluripotent cells, are called iPS. And we have two speakers, and as indicated in the program, we will ask our questions at the end of the session. And the first speaker is Jack Parent from the Michigan University, and he'll talk about the human neuronal model of PCDH19 linked epileptic encephalopathies. Thank you, Jack.
Okay. All right. Thank you. Thank you, Francesca, for putting together another great conference. It's really a pleasure to be here. And thank you to the audience for your endurance. So I'm going to kind of change paces and talk about human pluripotent stem cell model of PCDH19. And I think Joseph after me is going to do the same thing. So we've been using human pluripotent stem cells to model genetic epileptic encephalopathies since about 2009, starting with some of the ion channel epilepsies. And there are some advantages of using human pluripotent stem cells over mouse models and other approaches. If you get the cells from patients, they already contain the patient mutation on a human genetic background. There are a lot of issues with mouse background affecting seizure phenotype. You can make an unlimited supply and theoretically any cell type that you want to study. And you can use gene editing in two ways. You can correct a mutation to make an isogenic control, or if you can't get patient material, you can put in the mutation with CRISPR gene editing to make a virtual patient. And finally, you can use 2D culture assays, but more recently, a number of groups have developed 3D cerebral organoid assays, so-called mini brains in a dish, where the brain develops in suspension and gets closer to human brain organization. And I'll briefly talk about that. So our goal is to look at patient and control induced pluripotent stem cells, generate excitatory cortical neurons, inhibitory neurons, mixed cultures, look at protein and gene expression, electrophysiology with patch clamp recordings, and then multi-electrode array recordings that have been introduced in some of the earlier talks. Okay. So C.C. Du, a former grad student in the lab, started this project generating induced pluripotent stem cells from 3 patients with PCDH19 linked epilepsy. One is a point mutation, one is a large deletion, and one causes a frame shift and premature stop codon. I'm not going to talk about patient 2 because I think this patient, because of the deletion, had skewing and behaved like a wild type. I think it preferentially inactivated the mutant. But we have evidence for patient 1 and 3 that both alleles are expressed in the cultures. So CC showed that her stem cells expressed pluripotency markers, had normal karyotypes, and this is an example of a line that shows a single exist cloud, suggesting X inactivation in the pluripotent stem cells, but we have many other lines where we don't see the cloud and have reactivation. And in part, that depends on the culture media that you use. Some things such as lithium in the culture media may produce reactivation of the X chromosome. And that makes it difficult to study this disorder because in your cultures you never know how much X, normal X versus mutant X is expressed in any given culture experiment. So for that reason, the other thing we did is use a male carrier from patient 3. Actually, that was on the previous slide. So the father of patient 3 donated a skin biopsy, and we generated pluripotent stem cells. And then we used CRISPR gene editing of a normal wild type line to make lines with 2 and 5 base pair deletions that were virtual patients. So these are male cells where we knocked out the only X chromosome PCDH19 gene. And if you look at expression of PCDH9, starting with primers from the 5' or 3' end, the male CRISPR knockout has very low levels compared to female control. And interestingly, the male control has less than the female control, suggesting that the female may have both X's active and be transcribing. And we looked at another X-linked gene, HPRT, and we see a similar effect where there's more in the female. We don't entirely understand that. So you've seen this versions of this diagram of cellular interference. This was my grad student's version, where you have the normal state with wild type or carrier male, but with heterozygous female or mosaic male, you get segregation. And she colored these in 2 different colors because what's nice about this in vitro model is you can do mixing experiments like what Paul showed with his cell line. And I'm going to show some experiments where we mix the knockout cells with control or the carrier male cells with control. Okay, so to generate excitatory, so I'm going to focus on excitatory neurons today. So to generate excitatory cortical neurons, we use a dual SMAD protocol from Shi and Livesey, which makes neural rosettes, and with further differentiation, you get maturation of neurons. And the rosettes look like this. So these are stained for PAC6 in green, nestin in red, and a nuclear stain in blue. And you see these nice neural rosettes that have lumens. And when we compared the patient to the control, we see a nice rosette. And the lumen is analogous to the apical lumen of the neural tube and ventricular zone. And this stains for PKC, gamma, and cadherin. And with the control, we see a nice tight lumen. And with the patient, there's disruption of this lumen with both PKC, gamma, and cadherin expression. And I think Joseph probably sees the same phenotype in his cells that he might talk about. Now, interestingly, the carrier male, so when we mixed carrier male with carrier male cells, everything as expected was normal. When we mixed the carrier male with control cells, the PKC gamma didn't look very disrupted, but the N-cadherin did. So it was kind of a partial phenotype that we don't understand so much. In these experiments, we didn't label the mixed cells, so we don't know the proportion in the rosettes, but I'll show you some other mixing experiments where we did label the cells. Okay, so this is, these are the neurons from one of the patients, and all of the neurons in both the control and the patient do show a single exist cloud and H3K27 trimethylase labeling, suggesting that even if there was reactivation in the IPS cell line, when they are differentiated in the neurons, they do inactivate the X. And when we sequence, we find mixed populations in the culture, so there's expression from both alleles. And when we look at neuronal morphology, labeling with an immature neuronal marker double cortin or a more mature marker MAP2AB, the controls have normal morphology, and we see that the patient cells kind of don't like to migrate and spread out and kind of remain clustered, strangely. Might have an abnormal morphology with crossing of processes along itself, but that's still not entirely clear. And we think that they mature faster. When we look at a deep layer marker like TBR1, it's expressed in both the control and the patient. When we look at deep and superficial marker, SATB2, we see the deep marker in the control, but we don't yet see the SATB2, whereas at this time point, we already see both markers in the patient. Typically in the control, this will come on with further differentiation. So we wanted to look at function, and so we used multi-electrode array recordings, and these are plates from Axion, a commercial system, that make 12, 48, and 96 well plates. The 96 well plates have 8 electrodes per well, so you can do a lot of recordings. You get raster plots like this. You get real time. I'm not sure if this movie's going to work, but we'll give it a try. There we go. So you see the activity, and this is actually bursting activity. This is not from PCDH19. It's just an example from a different epilepsy gene mutation where we see bursts. And then you can get heat maps of the activity. Oop. This may or may not work if I can find the pointer. There it is. Yeah, this one didn't want to work before either. Anyway, so this is just showing more or less activity in the different wells. And when we compared one of the patients with control and looked at mean firing rate, it was markedly increased in the patient. This is days after plating, so it's still going up at 35 days after plating, and it's higher than the control. This is firing rate's probably not the best measure in these multi-electrode array recordings, but when we look at bursting, we see increased burst number and burst frequency in the patient, no change in burst duration or number of spikes per burst. And then you can also look at network bursts. So these are electrodes in the same well that are bursting at the same time, and we see increased network burst frequency and duration, as well as more spikes per network burst. So altogether, it's suggesting a hyperexcitability phenotype. The caveat is, so far this is only one patient and one control, and there's a lot of variability with these MEA recordings, and in fact, for other diseases, we've turned to making neurogenin 2 induced neurons, which make a more homogeneous population of excitatory neurons, and that limits the variability. So I think that's the direction we're going to go for this. Okay, so I mentioned the mixing experiments. So here we mixed the CRISPR knockout control. I'm sorry, we mixed a male control with the carrier male and compared it to an experiment where we mixed the male control with the male control or the carrier male with the carrier male cells. And we labeled each population red or green. And when carrier male and carrier male are mixed, you get mixing in the cultures and the morphology looks normal. When the controls are mixed, it's the same thing, but with mixing of the control and the carrier male, we saw a lot of colonies where only one type of cell was here, in this case the carrier male line. And the morphology, again, looks distorted with the processes kind of twisting on itself and very different than here. So this is suggesting that we can recapitulate that segregation phenotype in vitro. Our goal is actually to do it in the cerebral organoids, but we haven't done that experiment yet. So, and then taking a cue from Paul Thomas, he helped talented research scientists in my lab, Wei New, with some advice, because we wanted to tag the PCDH19 gene in the iPS cells, or in this case, in H9 human embryonic stem cell line. And so the idea is to make the HA flag tagged line so that we can identify expression and localization, but then to also do a CRISPR knockout of the other allele and be able to know which cells express the mutant and or have the loss of expression, which cells have the tagged line. And so Wei started the first part to make the tagging with homology-directed repair in CRISPR with Cas9 and the guide RNA, and put it in the C-terminal region. And when she looked at the lines, and she has some lines that are both alleles are tagged and some lines where only one allele is tagged, and compared it with the wild type, she sees no difference in gene expression. And interestingly, when we just look in general in the iPS cells at PCDH19 expression, we see compared to the undifferentiated cells, the rosettes have the highest expression, and then it's still high in the neurons, but so much higher in the neurons than in the undifferentiated pluripotent cells, but not as high as in the rosettes, which fits with Paul's data from mouse that the kind of ventricular zone stem cells have the highest expression. So this is examples of the tagging. So below is the wild type, and on top is a homozygous tagged line. And this is HA, which you don't see in the untagged, and you see nicely in the apical domain of the rosettes. And here's the flag and the merged image. And that HA tagging of the rosette lumens corresponds to N-cadherin labeling as well. So we think the tagging's working, and it's expressed in the correct domain, and it's a place where we would expect the phenotype that we saw. So now we want to look in cerebral organoids, and there are about 5 or 6 different ways to make cerebral organoids, and we've done 3 or 4 of them. Here I'm showing you Sergio Pasca's method, which he calls cortical spheroids. And basically, you use patterning molecules, the SMAD inhibitors, and growth factors, as well as neurotrophins. And they're growing in suspension and forming kind of brain-like structures that have a ventricular zone that expresses PAC6 and a deep cortical layer expressing CTIP2. Here's beta tubulin, so you get lots of neurons. This is at 5 weeks. By 11 weeks, you see lots of MAP2AB positive neurons, and many of them are expressing deep layer markers. With this, you usually get both SATB2 superficial cortical layer and CTIP2 deep cortical layer. So Wei looked at expression in the organoids. This is at day 46. So these are control with no tag. You don't see any HA labeling, but here you see a nice rosette structure expressing HA. And then we also got the antibody from the Japanese group that published it recently in Scientific Reports. And you see co-localization of PCDH19 and the HA tag in the rosette structure. This is at 46 days, and here at 76 days, it's in the rosettes, but it's also looking like it's in the migrating neurons, as one would expect. And it overlaps very nicely with the PCDH19. So we've seen a few phenotypes in the patient-derived iPS cells: abnormal rosette structure, premature differentiation, possibly abnormal morphology, and hyperexcitability with the MEA recordings. The mixed wild type and isogenic cultures have a partial rosette phenotype, but they show evidence of segregation in 2D cultures. And our goal for the future is to do the mixing experiments in the cerebral organoids, use the gene editing of the tagged line to make it a virtual patient line, and then generate inhibitory neurons. And there are ways using the Pasca method to make subpallial spheroids and cortical spheroids and fuse them together, where the interneurons migrate from the subpallial into the cortical spheroid, just like ventral to dorsal migration in normal brain development. So with that, I'll end and just thank the people I mentioned who did the work. Cece started it. Wei Nu made the tagged line. Sandra helped her with the cultures. And Andrew Tidball did the combined. So when we did the CRISPR knockout, we did it at the same time that we reprogrammed the fibroblasts, which is a really efficient way, and he developed that method in our lab. And we want to thank funding from Francesca's group, as well as Acute Syndrome Foundation. So thanks for your attention.